Good morning or good afternoon, whatever the case may be. Hey guys, this is Mr. Garner, and we are going to go ahead and get started with our instruction for today. Let's go ahead and learn about antibody production and vaccination. Go to view. Okay, so this is topic 11.1. We have completed the other topics thus far. In 11, we've completed 11.2, muscle movement. We completed 11.3, which is osmoregulation and ultrafiltration, and the kidneys and the nephrons. And we have completed 11.4, which was sexual reproduction and all of that fun gametogenesis. So now we're going to talk about 11.1, which is antibody production and vaccination. Let's get started. Okay. The first thing to understand about any organism is that that organism protects itself using its tags on its cells. So in every single cell, right, we have glycoproteins that are on the surface of the cell membrane, and those glycoproteins help to identify any of the cells within that organism. And that organism protects itself and maintains homeostasis from intruders from outside via self-recognition. So all organisms can recognize their own cells and recognize foreign cells that are trying to invade them. This is fairly important, as you can imagine. If you can imagine like a city with a lot of soldiers in it, it would be really good if that city could recognize soldiers from other cities, so they didn't just let them walk right in and be like, hey, come right on in. The other soldiers are like, oh, that's great, thanks. Now we will take over your city. And the first soldiers are like, man, we really should have read that book, Soldiering for Dummies. Anyway, so recognition of self and destruction of foreign. How does this happen? So all nucleated cells, basically any cell with a nucleus, have these major histocompatibility complexes. And these things are camping out on the membrane of all the cells in the body, and they all help to identify that body as you. So, if we think about in Burgess's cells, each one of Burgess's cells has a little major histocompatibility complex tag that's like Burgess's cell. And the same goes for Nyasha and for Skylar and all of you. So you all have your own special little MHCs. So these MHCs, as you can see, are portrayed here, are different shaped molecules. Of course, they won't quite be quite as biker gangish or boring as these are, but you can kind of see that this is how the cell is able to mark itself. But who comes along to figure out whether or not they are friendly or foreign? Who is scanning for all of these little markers? Well, who is scanning are lymphocytes. So antigens, which are foreign substance, and MHCs are both scanned by lymphocytes. Lymphocytes, you've learned about these. These are like white blood cells. They're attack or defensive cells that help your body in its immune response. And they're going to go around and they're going to bind to all the cells around with their little receptors. And they're going to bind to something called the epitope, which is the little exposed portion of these molecules. And they're going to read it and scan it to see if it really does say Burgess's blood cells. If it's in Burgess's body and it says Burgess's blood cells, that blood cell is good to go. But if it's in Burgess's body and it says intruder blood cell, then the lymphocytes will trigger antibody production and serious butt gets kicked. So when I say antibody production, what do I mean? Antibodies are things that help to destroy pathogens, and they do this using a cool 5-step method called panic, right? I mean, we call it panic. It's not really called panic, but anyway. So we have a couple of different ways that pathogens can fight against these, or excuse me, antibodies can fight against pathogens. And you'll notice that each of these ways really is just exposing the pathogen and or making it difficult for the pathogen to do its job, while the actual attack molecules like lymphocytes come along to do the real physical damage. So antibodies, I don't want you to think of them as being like the actual attack guards. I want you to think of them as being the alarm that triggers. And the alarm that triggers is going to alert the guards of the presence of the enemy, but it's not actually going to do the destroying itself. So let's take a look at each of these. So precipitation. If you guys remember, if you ever did the DNA extraction, one of the last parts of the DNA extraction in a strawberry is when you add the alcohol into the solution. And the reason you do that is to make it so that the DNA will actually appear in the solution. Up until that point, because DNA is a polar molecule, DNA will have been dissolved in whatever solution you're looking at, and essentially it will be undetectable. When you add the alcohol, boom, suddenly DNA precipitates, which is what we say when we say that it comes out of the solution and it becomes clear that there is some molecule in that solution. So that's what these antibodies will do when they precipitate. They will bring soluble pathogens out into the solution, and this allows them to be recognized more easily by the body, and it messes up their binding properties so they can't as easily carry out their tasks. Agglutination we've actually seen before. I believe it was with this class that I did the agglutination lab. If it wasn't, I apologize, and we'll see if we can do that when we come back. But agglutination is really just a clumping response. When the antibodies bind onto the surface of the pathogens on their receptors, they kind of make all of the pathogens start to clump up together. And that, of course, makes it very difficult for the pathogen to do its job when it's like stuck to five of its buddies and it's like, help, right? So agglutination. Neutralization. Remember that everything in molecules has to do with the binding sites. If you can bind to something else, you can make reactions happen. You can synthesize molecules, you can break them down, you can do anything. But if your binding sites are destroyed, you really are screwed. I mean, there's nothing you can really do. It immobilizes you. Think back to enzymes, right? If we mess up the active site of an enzyme, there's nothing you can do. So neutralization, that's what we're going to do. We're going to block those dangerous binding sites to make it so that the antibody, or excuse me, make it that the pathogen, which is the dangerous thing, is unable to do its job. Okay, and then we have inflammation. So inflammation inflames the body around the infected tissue. How is this helpful? Because I think pretty much any time we've ever gotten sick and you felt like that inflammation, you felt the aches and pains, you've been like, and this is just suffering. This is suffering. This is not helpful whatsoever. But inflammation is actually really helpful. So inflammation can help to raise the body temperature around the infected area. This is also having to do with your temperature raising when you get sick. And this can help to denature some of those pathogenic proteins. Inflammation can also trigger the release of certain other hormones, which are signaling hormones that go off and be like, hey, help, right? So inflammation is an important piece of this. It's a physical way of your body to react to pain and to send pain signals down neurons to release those hormones. So while it sucks, it is a way of your body calling for help. And then finally, complement activation. So there are these wonderful proteins called complement proteins, but instead of being like, oh, that's a beautiful dress, or wow, you look really nice today, they rip you to pieces. So yes, they're a little bit opposite of what their name is, but complement proteins go in and they start breaking the binds between the amphipathic plasmolipids in the membrane and breaking them apart so that cell just falls apart. So instead of complementing them, they're like, haha, you will die now, which is great. Anyway, so this is how antibodies work. And remember, they are triggered by the lymphocytes, and the lymphocytes are triggered by the pathogens. There's a chain going here. Anyway, let's continue. Okay, so how do you tell if something is an antigen? Well, let's just say that we have some suspicious surface molecules that don't match the MHC. So you're looking around at that tag and you're looking, it's supposed to say Burgess cells, but instead it says intruder cells, and you're like, that doesn't look like Burgess to me. Another way is that if a molecule shows up where it's not supposed to. So let's say a virus comes in and that virus releases its DNA into the cytoplasm of the cell. Well, the cell is going to be like, what the heck is DNA doing out here? And that cell is going to be like, well, that DNA is very suspicious because DNA is only supposed to be within the nucleus of the cell. So the body will usually attack it and ask questions later, which is why it would really suck if some of your actual DNA made it out into the cytoplasm of your cell, in which case it would be promptly destroyed. That cell would probably die. Okay, and then we have molecules like large food molecules. So these are not actually pathogens per se. They're just in your body, and your body maybe didn't break them down properly, and they register as being baddies. So your body will destroy them ruthlessly, and the components will be reused. So basically, they're like the undigested remains of metabolism, I guess you could say. But they get metabolized anyway. When they get destroyed by the lymphocytes, it's this whole thing. Anyway. Sometimes they can register as being baddies as well. Okay, Now, antigen detection is awesome because it means that you can survive when you need to survive and keep out all the foreign molecules from your body. But it also means that if you're in trouble and you desperately need something like an organ transplanted from someone else, or if you really need blood, that means that you are in a world of hurt because if that person is not genetically close enough to you or they don't have those right MHC molecules, then you are basically screwed. Yes, sir. Oh, what time should I leave? Okay, I'll be out of here by then. Thanks. So you want to make sure that you are receiving it from the right place so that those MHCs can actually register as yours. So what's really cool about blood cells is that they are enucleated, which means that they don't have a nucleus. That means that they don't actually have those little MHCs. But a cell should not go around your body without some form of identification. So they do have something that's called antibody tags or markers. These markers are basically just glycoproteins that help to, identify that they are employees of the building even if they aren't nucleated. And so we have a couple of different types. We have glycoproteins A or B. They can either be alone or in combination with each other to make AB. And if there are no glycoproteins, no identification at all, which can happen, then they are O. Now, I just said that shouldn't be the case. Well, in an O individual, that's fine. It's just going to really send up red flags when that O individual's. Actually, no, that's not right. I'm so sorry. Sorry, O's are the universal donors because the body doesn't recognize that because they don't have any of those tags. Wow, this is great. This is going so well. Let's start over. Glycoproteins could be AB or AB in combination, and that's really important to allow the body to know what they are. But O's are like the secret agents that come in and they don't have any identification, but they're so beautiful, like James Bond or something. Nobody asks them any questions, and they're able to sneak right on through. I am so sorry. I am tired. So right now I am just exhausted. I've been doing this all day and my brain flatlined, so I apologize. O's are able to be donated to anybody because they don't have glycoproteins. Haha, they can sneak in under anybody's security. But then there's also the RH factor. So it's a secondary class of glycoproteins that is or is not present, positive or negative, whatever the case may be. And it can be in combination with A and B. So it could be A positive, B positive, A negative, B negative, AB positive, all that stuff. You can even be O positive and negative, which means that an O negative person can't receive from an O positive. So there's like all of this really complicated rules. It's actually very simple once you write it all down, which is really what I'm going to ask you to do in just a second, is to just use these rules to figure out which one would be transferable to which one. So we're going to do a little bit of practice here. You'll go back to the assignment on the Google Classroom, and it will describe to you how all of this works. And I want you to see if you can figure out who can donate blood to who and why. All right, so go ahead and do that. Okay, I will assume that you did that and that you paused the video and that you are using the honor system here and not just using this diagram to complete that activity unless you really need it. But please practice independently of using help like this on the diagram because it will help you a lot more than you just staring at the answers and putting them down. Okay, so now you know who can donate to who. But okay, so what happens if you do happen to get the wrong blood? So let's say that the Red Cross was being less than on top of things and they give you blood that was not your type. Well, then the opposing antibodies in your body will bind onto the surface antigens of the now markedly foreign blood that has entered you. It will cause agglutination and then hemolysis as the lymphocytes come and help to rip apart your blood cells, which is not what you want. Remember that blood cells, their whole job is to carry oxygen through your body and other nutrients. So when your blood cells start to get ripped apart, it really sucks for you. And if you're wondering, yes, it's only ripping apart the foreign blood cells that you received in the transplant, but if you were in the situation where you received a blood transplant, I'm just gonna take a wild stab and say that you really needed those. So you're probably still in a desperate lot of pain and you really, really need help. Okay. So continuing on, hemolysis, bad. Surface antigens, good. Opposing antibodies, good, if you get the right kind of confusion. Okay, so what is a what, what is a pathogen? A pathogen causes disease. Okay, so anything that causes disease. You can see that list is very long. I'm not going to go into each one of those. You know what a disease is. You've been sick before, and it's why we're not meeting today. So anyway, a disease is something that screws up your homeostasis. You have had your homeostasis screwed up a lot before, I'm sure, if you've ever gotten sick, or if you have coronavirus, which if you do, my sincere condolences to you. Not condolences as in you're going to die, but that just really sucks, and I really hope you get better. Anyway, so that's what a disease is. It's a very temporary thing. An illness, on the other hand, is chronic. Okay, so if you get a disease that leads to a chronic illness, like such as HIV that leads to AIDS, that's going to be a big problem, and it's going to really impact your life for the rest of your life. So that's what we mean when we talk about an illness. So disease can lead to illness. That is the way this terminology works, and that'll come back around, of course, later on in this course. Okay, so pathogens are evolutionarily specific to their hosts. So not surprisingly, they have evolved alongside their hosts for thousands, if not hundreds of thousands or millions of years, and they have become adapted to the particular surface receptor proteins on that particular host. If a virus is to interact with a host without those complementary receptors on the surface, then they will not be able to infect them. So interestingly enough, pathogens, much like poison, are relative to whatever organism is interacting with them, which I think is kind of cool. So some pathogens can, however, sadly cross from one species to the next, and actually, believe it or not, this is where we get major problems. So, we were all scared of swine flu, which is the passage of influenza from pigs to humans. We'll be taking a look at that in a minute. And we also were really afraid of a bird-based flu, which is what the Spanish flu was. And the reason why is that those flus or viruses or pathogens that have evolved to another host, we have no immunity built up against them. Our system will not be able to recognize them in time because remember that recognition, that evolutionary history goes both ways. Sure, if they evolved with us, pathogens can infect us, but also if they evolved with us, our bodies know how to recognize them. If they didn't evolve with us, we don't know how to recognize them. So if a flu is able to jump from a bird to a pig, and then from a pig to a human, because pigs are more evolutionarily related to humans than birds, that genetic distance, that narrowing of the genetic distance in the infection of a person with a bird flu-like virus could be devastating. It could cause a pandemic that would sweep the nation and the world, and we would have very little resistance or immunity to it, if any at all. So that's where we get these really dangerous pathogens. Now, in terms of coronavirus, I actually really, this is terrible as a biology teacher, but I haven't really looked into the evolutionary history of coronavirus, but extra credit for whoever is able to look that up and tell me where it came from, its evolutionary history, and yeah, that'd be really cool. I will give you extra credit for that. In fact, I will give you a 100 quiz grade if you can give me that information, and just send that to me via my email. Okay. So anyway, diseases that are passed from animals to humans are called zoonotic or zoonoses. No, it's not zoonoses. You can pronounce it that way, but I'm just letting you know it's not zoonoses, even though it really should be. Okay, so how do you get a disease? Well, this is the stuff I really don't feel like I need to talk to you about because I'm pretty sure everybody has actually had this experience. So I'm not going to say anything about this slide. I'm going to let you figure it out. You can read it. You can pause the video for yourself. Go for it. But I do want to show you this awesome graphic, which is hilarious. I love this, and I love whoever made this. This is hilarious and amazing. So yes, this graphic is just great. I really want this graphic now to just be somewhere. I don't know where I want it to be, hanging up on the walls of work somewhere, but I think this would be a great graphic to inform people of how to prevent the spread of disease. But anyway, so now, okay, so in the Google Classroom, that's it for notes. On the Google Classroom, I have posted an article for you to read and questions for you to answer. And I've just noticed about halfway through this, I noticed that I had not been bolding all the words and the terms. So what I talked to you guys about and the specific things I pointed out was the important stuff in here. But I mean, you just use your note-taking skills, be discerning. I will be better on the next one. I apologize again, but I am just exhausted. So, without any further ado, go do that. That's your first daily lesson. I hope you enjoyed it. Have a wonderful day and stay safe, stay well. Yeah, peace out and all that stuff.